Genetic code. So in previous classes we have studied what is mRNA. If you remember, M stands for messenger RNA, and I told you it is the RNA which carries the genetic information required for a polypeptide chain synthesis, and mRNA carries the genetic information in the form of codons. And if you ask me what is a codon, my answer is codon is nothing but triplet sequence of nitrogen bases. So triplet sequence of nitrogen bases is called a codon, and each codon codes for a particular amino acid at the time of polypeptide chain synthesis on ribosomes. So what is a codon and how many codons are present? Which codon codes for which amino acid? It was all a big confusion and a big mystery in the field of molecular biology, and this confusions and this mystery was revealed by the discovery of genetic code. So who discovered this genetic code and why the genetic code must be a triplet? All such things let us discuss in today's class. And simply if you ask me what is a code, in very simple terms, general definition for code is that code is collection of symbols which carries the information. So that is the simplest definition for code. So any code can be, it is a collection of symbols and those symbols carry some information. So in genetic code, it is the collection of symbols. What are those symbols we are talking about? They are nothing but nitrogen bases. As you all know, nitrogen bases are adenine, uracil, cytosine and guanine, because here we are talking about RNA which contains codons are always present on mRNA. In RNA, thymine nucleotide is not present. Thymine is replaced by uracil. So that's why I have written. So the four symbols which code for, that is adenine, uracil, cytosine and guanine. They are arranged into what I said, triplet sequence of nitrogen bases. So this triplet sequence of nitrogen bases is called a codon. What is the function of a codon? Codes for an amino acid at the time of polypeptide chain synthesis. And here, the first question, triplet sequence of nitrogen bases. This itself is a big question. Why code must be a triplet? Because why code must be a triplet? To answer this question, first we have to know the number of amino acids or the types of amino acids required for protein synthesis is always 20. So in any organism, for the synthesis of a polypeptide, how many types of amino acids are required? 20 amino acids are required. So that's why 20 amino acids are said to be standard amino acids. With this it is very clear how many nitrogen bases or how many types of nitrogen bases are present on mRNA. Only 4 types. So these 4 types of nitrogen bases must code for minimum 20 amino acids. With this it is very clear, based on permutations and combinations, number of codons must be minimum 20 or more than 20. So if you understand this statement, number of codons must be minimum 20 or more than 20. Why, tell me? Because minimum number of amino acids to be coded is 20. So with this idea, the concept of triplet genetic code or the hypothesis of triplet genetic code was given by George Gamow. So all students should remember, the bold hypothesis of triplet genetic code was given by George Gamow, because before this hypothesis many scientists were telling every nitrogen base acts as a codon. Just imagine, if every nitrogen base acts as a codon, how many types of nitrogen bases are present? Only 4. 4. So 4 types of nitrogen bases. If every nitrogen base is acting as a codon, so it should be what? 4 power 1. So they can code for how many amino acids? Only 4 amino acids. But how many amino acids is the minimum requirement? 20. So that's why 4 power 1 is not a possibility. So students should understand, number of codons should be minimum 20 or more than 20. If every nitrogen base acts as a codon, total how many codons can be formed? 4. So 4 is less than 20. So this is not a possibility. Next, let us consider code is a doublet. Code cannot be a singlet, it is proved. So let us consider code is a doublet. If code is a doublet, 4 power 2, how many codons can be formed? Only 16. Once again, 16 is less than 20 because I already told you number of codons should be minimum 20 or more than 20. So ultimately the only possibility is the very next possibility, 4 power 3. So 4 power 3 is nothing but 64. So 64 is more than 20, right? So based on this idea, based on permutations and combinations, as only 4 types of nitrogen bases has to code for minimum 20 amino acid, George Gamow has proposed the bold hypothesis of triplet genetic code. So according to this, in the genetic code library, totally how many codons are present? 64 codons are present. Out of these 64 codons, 61 codons code for how many amino acids? 20 amino acids. So 61 are called coding codons and they code for how many amino acids? 20 amino acids. And remaining 3. So 3 codons are called stop codons as they do not code for any amino acid and they stop the polypeptide chain synthesis. That's why 3 are called stop codons and 61 codons code for 20 types of amino acids. So altogether in the genetic code library, what is the total number of codons present? That is 64. Out of 64, 61 are the coding codons. They code for 20 types of amino acids and 3 codons do not code for any amino acid and such codons are called stop codons, which are the 3 stop codons etc. we discuss later. So first let me repeat and let me tell you what is genetic code. It is the collection of symbols. What are the symbols I am talking about? They are nothing but nitrogen bases. What information do they carry? They carry the information required for the synthesis of a polypeptide and they carry the information in the form of what? Triplet sequence of nitrogen bases. What do you call a triplet sequence of nitrogen bases? Codon. What do? They code for amino acids. And why code must be a triplet? Why can't be genetic code is a singlet or doublet? It was revealed by George Gamow. According to George Gamow, based on permutations and combinations, as minimum number of amino acids to be coded is 20, number of codons should be minimum 20 or more than 20. But how many types of nitrogen bases has to code for 20 amino acids? Only 4. So that's why if you consider code is a singlet, only how many codons can be formed? 4. So 4 is less than 20, it is not possible. Let us consider code is a doublet, 4 power 2, that is number of codons is 16. 16 is less than 20, it is not possible. So finally code must be a triplet. So when it is 4 power 3, 4 types of nitrogen bases arranged in triplets. So how many arrangements or how many combinations are possible? 64 combinations are possible. They are nothing but the codons. So 64 is more than 20. So happily they can code for 20 amino acids at the time of polypeptide chain. The concept of triplet genetic code was supported and further strengthened by So I already told you the bold hypothesis of triplet genetic code was given by George Gamow. So this triplet genetic code concept was supported and strengthened by Khurana, all-time great scientist in the field of molecular biology. So simply we can say the father of artificial gene synthesis. and he is also involved in the discovery of cloverleaf structure of tRNA. So a very great scientist in the field of molecular biology, Hargobind Khurana. He strengthened and experimentally proved that genetic code must be a triplet. So how he proved that genetic code must be a triplet? He synthesized an mRNA by making use of only two types of nucleotides, that is containing nucleotides are of what? Uracil and guanine. So he synthesized an mRNA artificially with the help of only two types of nucleotides. And if you see here, so if code is a doublet, means some scientists were arguing that code can be a doublet. If code is a doublet, how you have to read it? You have to read it as UG, 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 UG like that. So if code is a doublet, when this mRNA participates in translation, what is going to be synthesized at the end of translation? A polypeptide chain is synthesized. And if code is a doublet, if you read codon as UG, so all are same, that is UG, UG, UG like that. So in a polypeptide chain, what should happen? Tell me. Only one type of amino acid or same type of amino acids should be present in polypeptide chain. But when this mRNA was used for the process of translation, so at the end of the translation, an amino acid was produced, and this was having alternative sequence. So alternative sequence of what? Like this. It had alternative sequence of what? Cysteine, valine like that. So when a polypeptide chain was synthesized with an alternative sequence of two amino acids, that is cysteine, valine. So what does it mean? You should not read it as UG, 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 but you have to read it as UGU, GUG. Next once again, UGU, GUG, like this. So code must be a triplet. With this it is very clear. UGU codes for cysteine, GUG codes for valine. So if you consider code as a doublet, you have to read it as only what? UG, UG. And this polypeptide chain must have same type of amino acid. But when Hargobind Khurana synthesized an mRNA in the laboratory with only two nucleotides, that is uracil and guanine nucleotide continuously, when this mRNA was used for translation, a polypeptide chain with alternative sequence of cysteine, valine is synthesized. Then Hargobind Khurana said, you must read this code as a triplet, that is UGU, GUG, UGU, GUG like this. So with this experiment, triplet genetic code concept of George Gamow is proved once again, and from that point of time, there are no confusions in genetic code. Genetic code must be a triplet. And finally, we have to remember the name of Marshall Nirenberg. According to NCERT textbook, if the question is asked, who deciphered the genetic code? So answer should be Marshall Nirenberg. And today I told you, in the genetic code library, total how many codons are present? 64 codons are present. And out of these 64 codons, 61 are said to be coding and 3 are non-coding. Otherwise we can call them stop codons. And total 64 codons. Which are the 64 codons? And which 61 codons code for 20 amino acids? Which 3 codons do not code for amino acid? Everything is revealed, everything is deciphered by Marshall Nirenberg by studying cell-free system of protein synthesis. So students should understand who deciphered the genetic code. Answer should be Marshall Nirenberg. How he deciphered or how he explained the genetic code? By studying cell-free system of protein synthesis, means He studied the process of protein synthesis in the laboratory, not inside a cell. So that part of protein synthesis is called cell-free system of protein synthesis. By studying cell-free system of protein synthesis, Marshall Nirenberg deciphered the genetic code. Because of this only today we know which codon codes for which amino acid, which codons do not code for amino acids. Okay, and finally students should remember one more name. Severo-Ochoa enzyme. Severo-Ochoa enzyme is also called polynucleotide phosphorylase. And what is the use of this enzyme? So this enzyme is used for enzymatic synthesis of mRNA in DNA template independent manner. So it is very clear, whenever we talk about protein synthesis, mRNA is synthesized on template DNA. So with that it is very clear, for the synthesis of mRNA in living systems, DNA template is essential. But today in the laboratory we can synthesize an mRNA according to our own nucleotide sequence and or in defined nucleotide sequence without a DNA template. So if you want to synthesize an mRNA with a defined nucleotide sequence without DNA template or DNA template independent manner, which enzyme makes it possible? That enzyme is nothing but Severo-Ochoa enzyme. Actual name of this Severo-Ochoa enzyme is polynucleotide phosphorylase. So students should remember the important point. Enzymatic synthesis of RNA in DNA template independent manner is made possible by an enzyme called Severo-Ochoa enzyme. Other name for Severo-Ochoa enzyme is polynucleotide phosphorylase. So I want to stop this class by repeating. Here in this class, importantly you have to remember the name of George Gamow. So George Gamow has proposed the bold hypothesis of triplet genetic code based on permutations and combinations. Why George Gamow said that code must be a triplet? Because only 4 types of nitrogen bases has to code for minimum 20 amino acids. So if you consider each base acts as a codon, only 4 codons are possible. 4 codons can never code for 20 amino acids. So that's why he must set that code must be a triplet, which results in the formation of 64 codons. His simple idea was that as only 4 bases has to code for minimum 20 types of amino acids, number of codons should be minimum 20 or more than 20. It becomes possible only when code is a triplet. And this triplet genetic code concept of George Gamow was experimentally proved, supported and strengthened by whom? Hargobin Khorana. And finally you have to remember, directly if the question is asked, who deciphered the genetic code? We discuss genetic code library in the next class. In genetic code library, total 64 codons are present. Which codon codes for which amino acid? Today we know everything. That every information about genetic code library was revealed by Marshall Nirenberg by studying cell-free system of protein synthesis. And finally, I want students to remember, if you want to synthesize an mRNA without DNA template in the laboratory, today it is possible because of the presence of an enzyme called Severo-Ochoa enzyme, which is also called polynucleotide phosphorylase. What is the use of this enzyme? Enzymatic synthesis of mRNA with defined nucleotide sequence in DNA template independent manner. Thank you.